This video will examine the structure of nucleotides and explore some of the nucleotides' functions, particularly as components of DNA, RNA, and energy storage molecules. Every nucleotide has a pentose sugar, that is, a sugar molecule with 5 carbon atoms, and a phosphate group, and a nitrogenous base. The nitrogenous bases are either purines, which have 2 rings, or pyrimidines, which have a single ring. Now, let's look at these components more closely, starting with 2 sugars. One sugar is ribose, which is found in RNA and ATP. The other sugar is deoxyribose, which is found in DNA. The difference between ribose and deoxyribose is in the presence of an oxygen atom on the second carbon of ribose. This oxygen atom is missing in deoxyribose, thus the deoxy part of its name. In organic chemistry, carbon atoms are numbered. In nucleotides, they are numbered as 1' through 5'. This numbering, particularly 3' and 5', is very important, as we shall see. Now, let's look at the nitrogenous bases. The purine bases, each with 2 rings, are adenine and guanine. Both are found in RNA and DNA. Pyrimidine bases, with 1 ring each, are thymine, found only in DNA, uracil, found only in RNA, and cytosine, found in both RNA and DNA. Nucleic acids, whether DNA or RNA, are formed by stringing nucleotides together. This is where carbon numbering comes in. The base is attached to the 1' carbon, and phosphate is attached to the 5' carbon. A second nucleotide can be attached with its phosphate to the 3' carbon of the first nucleotide. Another nucleotide can be added to the 3' carbon of the second nucleotide, and so on. Because the beginning end of a nucleic acid has a phosphate on a 5' carbon, this end is called the 5' end. The other end of the nucleic acid is the 3' end. New nucleotides are only added at a 3' end. Besides nucleic acids, nucleotides are also used to make energy storage molecules of cells, such as Ch2p and the more common ATP. Here we examine the structure of ATP. In these drawings, the molecule is reversed from what we have seen. That is, the 1' carbon is on the left and the 5' carbon is on the right. A nucleotide without a phosphate is called a nucleoside. Many nucleoside analogues are used in treating diseases, such as AIDS and herpes. In this figure, we see adenine ribonucleoside. This is also called adenosine. Adding a phosphate group gives us an adenine nucleotide, which can also be called adenosine monophosphate, or AMP. Cyclic AMP is an important signal molecule inside cells. Adding a second phosphate group gives us adenosine diphosphate, or ADP. The bond between adjoining phosphate groups can be thought of as a high-energy bond, because the phosphates linked together have more energy than the sum of the 2 phosphates independently. Adding a third phosphate group gives us adenosine triphosphate, or ATP, which has another high-energy bond. ATP is the energy currency of a cell, acting as a sort of reloadable energy charge card. In summary, nucleosides have a sugar and a base, while nucleotides have sugar, phosphate, and a base. The bases are either the purines, adenine and guanine, used in DNA, RNA, and ATP, or the pyrimidines, which are thymine used in DNA, uracil used in RNA, and cytosine used in both DNA and RNA. The sugars are ribose used in RNA and ATP, and deoxyribose used in DNA. The nucleoside adenosine is composed of ribose and adenine base. Adding phosphate groups gives us AMP, ADP, and ATP.